Hello friends, Ace Botany next episode. In this episode, we'll be discussing photosynthesis fourth one. And what all things we'll be discussing in this episode of photosynthesis. For that I will share the screen so it will be convenient. I think screen came. So we can proceed. 4 items we'll be dealing, of which fourth one is question, 5 theory, 5 NEET model question with a key. Other 3 aspects, where does ATP and NADPH used, dark reaction, chemiosmotic hypothesis. So we can start. First is, where does ATP and NADPH used? Before it, we should know in which reaction it produces. Already we studied previously, lighter reaction, 3 products will be there, ATP, NADPH and oxygen, where oxygen will be diffusing out, ATP and NADPH assimilatory power that will be used to produce food, otherwise sugar, through specific reaction. And this reaction pathway is often called dark reaction. Automatically we can enter into dark reaction. Many synonyms are available, like carbon fixing reaction, light independent reaction, even Calvin cycle, biochemical pathway, or even biosynthetic pathway or phase. Light reaction is called photochemical pathway. Then another names of dark reaction is reductive pentose pathway, RPP. Then PCR cycle, photosynthetic carbon reduction cycle. This way many names are there. And this can take place in all photosynthetic plants, no matter whether it is C3 or C4 or even C2. Basic reaction will take in all such plants. And dark reaction not depends directly on light, but products of light absolutely need, mainly ATP and NADPH. That means name dark reaction can be a misnomer. I think up to that it is clear to all of you. Now we can proceed to C3 cycle. Famous scientist is Melvin Calvin. For this work, he received Nobel Prize in 1961. That is why in each episode of photosynthesis, if you check this photograph, you can observe this is the scientist Melvin Calvin. Now, before entering into the reaction side, we should know what is the product form and which organism is used, which methodology is used, which carbon is used, how the organism killed, then radioactive substances separated, all this. This aspect not given in NCERT. So carbon will be converted into carbohydrate. First stable product is a 3-carbon compound, that is 3-phosphoglyceric acid. That is why it is called C3 cycle. Suspension of chlorella will be utilized. Chlorella is generally used for physiological experiment. Large chloroplast is there. And autoradiography they utilized. Carbon 14 is the chemical. Half-life period is 5730 plus or minus. And the organism chlorella killed in hot methanol. Radioactive material separated by 2-dimensional paper chromatography. That is why we studied paper chromatography in first or second episode of photosynthesis. NCERT also given the term paper chromatography. Finally identified by autoradiography. An example for some C3 plants. Odd one question, mixing with the C4. Soybean is there, wheat is there, cotton is there, rice is there, potatoes are there. That is regarding the first page, hoping that it is clear. Now we can proceed to second page. Cycle. This is the NCERT diagram for C3 cycle. Before getting into it, we can observe this area. Main enzyme involved is rubisco, and it is seen in the stroma side, or the stroma on the outer surface of the thylakoid membrane. Synonym of this enzyme, ribulose 1,5-biphosphate carboxylase or oxygenase, rubisco. So carboxylase or oxygenase. Previous name, carboxydismutase. So main name is rubisco. Old name is carboxydismutase. It can do carboxylation, then carboxylase. It can do oxygenase, oxygenation. I think that part is clear. Now the substrate. One is carbon dioxide, one carbon compound. Now second is RUBP. RUBP is a 5-carbon keto sugar. And remember, Rubisco, most abundant protein in the biological world. 3 stages are there, and that stage is given here. See, 1, 2, 3. That itself is a question. 3 main reactions. First is carboxylation. Second is reduction. Here you can find carboxylation, reduction, regeneration. These are the compounds. In between compounds not mentioned. Anyway, 3 steps: carboxylation, then reduction, regeneration. That itself is a question. Reduction sometimes often called glycolytic reversal. Now, next area. See, original C3 pathway is not like this. This is only a skeleton. And that pathway is not given in NCERT book. One typical example I'll tell you. There is an enzyme in C3 cycle, namely aldolase. This enzyme is studied in glycolysis. So aldolase can be seen in C3 cycle as well as glycolysis. In C3 cycle, how it works, act as a ligase. 2 3-carbon compounds will be coupled to form fructose 1,6 biphosphate. While in glycolysis, same fructose 1,6 biphosphate will be cleaved into 2 3-carbon compounds, same enzyme, that is aldolase. So this way, so many things are there in original reaction cycle, but that part is not shown there. This is NCERT version. I think it is clear. And the detailed diagram I given in page number 7. When we reach there, I will show you. Now we can proceed to next page. Slightly expanded diagram I show. Again, it is not complete, but still skeleton little bit expanded. We can find 6 molecules of carbon dioxide that is taking part of coupling with 6 RUBP. RUBP is a 5-carbon compound keto sugar. So carbon dioxide coupling, so carboxylation enzyme is Rubisco. That is why carboxylation. And we'll be getting a 6-carbon, 6-molecule compound. It is highly unstable. Name of the compound is 2-carboxy-3-keto-ribitol 1,5-biphosphate. And soon it will be converted into 12 molecules of a 3-carbon compound, phosphoglyceric acid. That is why first stable product in C3 cycle is 3-carbon PGA. That is why the cycle is called C3 cycle. 3 PGA will be converted into 12 molecules of di-PGA. So phosphate has to be added. 12 molecules will be added. This ATP will be produced in light reaction, 12 ATP. Now, 12 molecules of di-PGA will be converted into 12 molecules of PGAL, acid to aldehyde. Reduction. And reduction power comes from NADPH. In light reaction, we'll be remembering 12 NADPH is already there. 12 PGAL will be cleaving into 10 PGAL and 2 PGAL. That is why reduction, after that it is otherwise called glycolytic reversal. That is formation of food or sugar. What is the fate of this 10 PGAL? 10 PGAL will be going back to produce RUBP. 6 molecules of RUBP will be produced. For that 6 more ATP needed. So 12 here, 6 here, 18. And that event is called RUBP regeneration. Again, 6 molecules of carbon dioxide will be coupled. Cycle will be get repeat. I think it is clear. After that, a statement with the green color I mentioned. 3 steps. Carboxylation is the most crucial step. Step number 2 is reduction, or sometimes called glycolytic reversal. Third one is regeneration of RUBP, that is 2, or for the continuation of the reaction. So Rubisco and other enzymes in C3 cycle are regulated by light through different metabolism. That aspect we are not discussing in NCERT. Now, chemicals used. Here we can find 6 carbon dioxide, 18 ATP, 12 NADPH. What we get is 1 glucose, 18 ADP, 12 NADP. I think that part is clear. Now we can proceed to famous theory, chemiosmotic hypothesis. So what is the meaning? That meaning already we discussed in respiration since we finished respiration first. Same aspect. Basically, movement of ions across a semi-permeable membrane based on electrochemical gradient or difference in amount or concentration. Same theory also seen in respiration. Both side ATP has to produce. Scientist is Peter Mitchell, 1978, chemiosmotic theory, Nobel Prize he received for this chemiosmotic theory. Now we can proceed to next page. Here, ATP synthesis is connected with development of a difference or gradient of proton. Gradient means difference. One side it is more, other side it is less. Connected with the thylakoid membrane. Semipermeable membrane should be there of chloroplast. And there are several mechanisms available while activation of electrons and the transport will cause development of proton gradient. So proton gradient should be produced. Two areas, one side more proton, other side less proton. So how this proton gradient or accumulation takes place? NCERT explains three methods. NCERT. One, water splitting. So photolysis takes place towards thylakoid lumen. This diagram is the NCERT diagram. It is very, very clear, self-explanatory diagram. Everything marked clearly there. So here you can find water. Splitting of water, again protons accumulated in. This is lumen side. This is stroma side. So lumen. This will be the membrane. It's a very good diagram. With this, we can explain everything. So automatically photolyze of water, that will cause accumulation of protons in the thylakoid lumen. One method. Second method is, see, this is photosystem 2. This is photosystem 1. So originally it is non-cyclic photophosphorylation. So when electrons move through photosystems, protons also get transferred across the membrane. There is a reason for it. See, here you can find. Starting with the PS2. Primary acceptor of electrons, firefighting, which is located towards outer side of the membrane. So PS2, so somewhere here. Membrane transfers its electron to a proton carrier. So from firefighting, electrons will be transported to a proton carrier. Which is the proton carrier? Here you can find it, plastoquinone. And plastoquinone is a unique protein, is both an electron and proton carrier in photosynthetic membrane. And this molecule then receives H+. So now you can find this common PQH. So proton from the stroma, says stroma side. So proton from the stroma side, while transporting an electron, will be added. From PQH, molecules passes the electron carrier present in the inner side of the membrane. So you can find it is passing into plastocyanin inside the membrane. So lumen. H plus will be released to thylakoid lumen. CH plus. So very beautiful diagram. Now third one, NADP adaptations, and now we are proceeding to here. Is located on the stroma side of the membrane, this area. Along the electron that comes from the acceptor of electron PS1. So from PS2, PS1. Protons are necessary for the reduction of NADP to NADPH here. And these protons are also removed from stroma. So all these mechanisms will cause decrease of amount of proton in stroma. Stroma means this side. At the same time, protons accumulate in thylakoid lumen. So this will cause a proton gradient across the thylakoid membrane as well as measurable decrease in pH in the lumen. So proton increases. Proton. So decrease in pH in the lumen. That statement is given in NCERT pages 140 and 141, March 24 edition. And what is the importance of proton gradient? Proton gradient is a must in connection with the ATP or ATP production. Breaking this proton gradient, then only ATP will be released. So for this chemiosmotic theory, or here requires 4 items: membrane, proton pump, proton gradient, ATP synthase. And the proton gradient or gradient of proton is broken down through ATP synthase, famous enzyme. So that aspect now we can proceed. This all I given here, but to discuss with the diagram, I put the diagram page. That's all here. Now we can proceed to next aspect. This is the famous chemiosmotic theory. Then from there, that part is LR. So diagram is very clear after enlargement. This is F0, this is F1, this is stroma side, this is thylakoid lumen, everything, this is membrane, everything clear. ATP synthase, CF0, CF1, C4 chloroplast has 2 parts. CF1 is a catalytic part. CF0 is part embedded in the membrane. Now CF0 gives a channel or tunnel. See this one. From lumen to stroma. Proton will be migrating back. So tunnel or channel for the passage of protons while breaking concentration of proton motive force. While CF1, this one, project on the outer surface, see this area of the membrane seen in stroma side. This is stroma side. And it is catalytic in function, ATPase. Now proton migrate back to stroma. So from lumen, it comes back to the stroma by facilitated diffusion through CF0. This way it comes down. CF0 is proton tunnel or channel. Breakdown of this gradient provides enough energy to cause a conformational change or conformational change in CF1, this area, and CS1 of ATP synthase to cause production of ATP. So this diagram clearly explains everything, how this proton motive force is broken and how it comes down, which is the channel, how ATP produced by consuming or utilizing CF1, etc. Beautiful diagram. I think it is clear. Now, next is, I told you a real C3 cycle that will be in this form. Here you can find the famous hydrolase enzyme, which will be acting as ligating and lygating enzyme. After this, no need to study all these details because not in NCERT, just mentioning. Now, 5 theory question, 5 NEET model question with the key. So what all things we learned in this episode? We can just pass through. So we started with introduction, what all things we'll be learning. Then where does ATP and ADPH used? Then dark reaction, is it a misnomer? C3 cycle, Melvin Calvin, Nobel Prize. After that, some C3 plants, then the cycle here, mainly the chemicals. Then after that, 3 stages. Then this area, original representation I just mentioned. Then to the next page, C3 cycle, another form of representation. How many ATP, how many NADPH, and what is the importance of these steps? Carboxylation, most crucial. Regeneration of RUBP for continuation of the cycle. Then what all chemicals utilize, what all produce, and famous chemiosmotic theory. And that beautiful diagram explaining everything, how the proton accumulation takes place, water splitting, when electrons move through photosystems, and finally NADP reductase enzyme. So water splitting, photosystems, NADP, NADPH. After that, how ATP synthesis takes place. This diagram, CF0, CF1. CF0 gives a channel, CF1 that is acting as ATPase or catalytic function, facilitated diffusion, proton migrate back, and producing ATP. After that, a big representation. Then 5 theory question, 5 NEET model question with the key. If you like it, subscribe all, like it, share it, comment on it. We can meet with the next topic in next episode. Time being, we can wind up. Thank you.